Good morning. Today I will be presenting our research findings on immune cell modulation of pulsed focus ultrasound in murine melanoma and breast cancer models. Immune evasion techniques of tumors frequently depend on the aberrant crosstalk between innate and adaptive cell lineages, which could promote tumor progression and metastatic dissemination. Tumor microenvironment could be classified as having infiltration excluded cold or infiltration inflamed hot immune landscapes depending on the extent of CTL infiltration into the tumor bed. Our specific research interest was to see if mechanotransductive effects of non-ablative PFAS was capable of promoting immune cell infiltration, helping to shift a cold TME to an immune-active hot TME. Murine B16 melanoma or 41 breast cancer cells were subcutaneously implanted into C57 or BALB/C mice flanks, and once tumors reached approximately 5 mm in size, ultrasound-guided PFAS was administered at a peak negative pressure of 6 MPa. Tissues were harvested on days 1, 3, and 5 post-PFAS for various analysis. Why 6 MPa? Tunnel assay was performed across a range of peak negative pressures to identify an optimal PNP which could trigger TNA damage, a potent danger signal capable of releasing damp molecules which could initiate a pro-inflammatory response. Results revealed PFAS to generate significant DNA damage in both tumor types at 6 MPa, and thus this PNP was selected for our subsequent PFAS treatments. Also, H&E demonstrated no consistent pattern of increased hemorrhage or necrosis or any evidence of thermal injury in 6 MPa treated tumors compared to controls. Can non-ablative PFAS alter tumor progression or micromets in tumor tissue? We've been looking at these models harboring small-sized tumors as representatives of micrometastatic infiltrated disease in normal tissues, wherein non-ablative PFAS could stimulate an immune response without heating up the normal tissues where the micromets reside. Here, both tumor types exhibited significant reduction in tumor volumes following PFAS treatment over 5 days compared to untreated controls. This is the B16 flow data represented as heat maps, and looking at the tumors on day 1, effector immune cells and APCs are suppressed, and on day 3, we saw active immune cell influx with increased numbers for cytotoxic and helper T cells, macrophages, and NK cells, all of which decreased by on day 5, except for NK cells which remained high in all 3 tissues. In the case of 41, what started as a cold tumor on day 1, slowly by day 5 starts to heat up with influx of more cytotoxic and helper T cells, M1 macrophages, NK and dendritic cells, along with suppression of anti-inflammatory Treg, MDSC and checkpoint markers. We simultaneously looked at the CCTF expression in both B16 and 41 TMEs over 5 days, and both CCTF profiles showed increased expression of pro-inflammatory CCTFs capable of promoting an anti-tumor environment in response. This is the gene set enrichment analysis data for gene ontology of biological processes enriched 1 and 12-hour post-PFAS compared to untreated control. Nodes are clustered by similarity, and at 1 and 12-hour post-PFAS, different network patterns were observed for each type, wherein B16 predominantly showed upregulated immune responses, while 41 was characterized by downregulation of cell proliferation and tumorigenic processes. Also, calcium transport and signaling, which was upregulated in 41 at 1 hour, was negatively enriched in B16 at 12 hours. This is the heat map showing changes in gene expression for GSE enrichment of gene ontology hallmark gene sets that were differentially enriched by PFAS in 2 tumor types: hypoxia, epithelial to mesenchymal transition, KRAS, and calcium signaling enrichment scores following PFAS were significantly different between both tumors. So our results indicate that PFAS can indeed shift a cold TME to a hot one. Conclusions: this study reveals the differences in immune cell, CCTF, and transcriptomic profiles following sonegation between 2 tumor types and underscores the importance of not generalizing the effect of PFAS on TME. Both tumors exhibited significant reduction in tumor volumes. Temporal changes in the cytokine and immune cell profiles following PFAS demonstrated anti-tumor response by engaging both innate and adaptive arms of immunity. Transcriptomic analysis revealed differential and divergent expression patterns for biological processes between two tumor types, and the observed global change to the immune cell landscape represented a shift from cold TME to a pro-inflammatory hot TME. Thank you.